Let's now talk about the last but not the least component, the nucleotide. You can look at the image, it's the structure of a nucleotide, the basic unit that makes up DNA and RNA, and are really important because they help carry and pass on genetic information in all living things. So, this nucleotide is not just a simple molecule, it is the molecular blueprint that forms the very basis of life. The DNA molecule itself is made up of several key elements like carbon, hydrogen, oxygen, nitrogen, phosphorus, and sometimes even sulfur. These elements come together in a very specific way to form long, twisted chains we know as the double helix. DNA and RNA are built from long chains of nucleotides, and these chains are arranged into two strands in the case of DNA. So, what exactly is a nucleotide? We can say that nucleotides are the building blocks of DNA and RNA. Each one is made up of three basic parts. First, we have the phosphate group. This group contains a phosphorus atom bonded to 4 oxygen atoms, and it carries a negative charge. This part is important for linking the nucleotides together. Second, there's the sugar molecule. It's a pentose sugar, which simply means it has 5 carbon atoms. In DNA, the sugar is called deoxyribose, and in RNA, it's ribose. The difference between the two sugars affects the structure and function of DNA and RNA. And finally, we have the nitrogenous base. This is a molecule that contains nitrogen, and it's what makes each nucleotide unique. The sequence of these bases is what carries genetic information. So in short, every nucleotide is like a tiny puzzle piece made up of a sugar, a phosphate, and a base. Together, they form the long, information-carrying strands of DNA and RNA that make life possible. Now that we've understood what nucleotides are made of, let's move a little further to see how they actually function as the basic building blocks of nucleic acids, which include both DNA and RNA. To help us understand this better, let's first look at the types of nitrogenous bases found in these nucleotides. There are 2 main categories. First, we have the purines, which include adenine and guanine. Then, we have the pyrimidines. In DNA, these are thymine and cytosine. But in RNA, uracil replaces thymine, so RNA contains uracil and cytosine as its pyrimidines. Now remember, a complete nucleotide is formed when 3 components come together, a phosphate group, a sugar molecule, and one of these nitrogenous bases. Let's take a closer look at the actual components of DNA. In DNA, the sugar used is called deoxyribose, and the nucleotides are as follows. Adenine, combined with deoxyribose and phosphate, forms what we call deoxyadenosine monophosphate. Thymine, with deoxyribose and phosphate, becomes deoxythymidine monophosphate. Cytosine, along with deoxyribose and phosphate, forms deoxycytidine monophosphate. And guanine, with deoxyribose and phosphate, is called deoxyguanosine monophosphate. And, now moving to RNA, which uses ribose instead of deoxyribose as its sugar. In RNA, thymine is replaced by uracil. So the nucleotides in RNA are adenine, with ribose and phosphate, forms adenosine monophosphate. Uracil, with ribose and phosphate, becomes uridine monophosphate. Cytosine, with ribose and phosphate, forms cytidine monophosphate. And guanine, with ribose and phosphate, gives us guanosine monophosphate. These nucleotides act as the monomers, or the small repeating units, that link together to form long chains of DNA and RNA. And most importantly, the sequence of these nucleotides along a strand is what encodes genetic information, which directs all the activities within living cells. If you look at the image, you can see how DNA is tightly packed inside the cell. This shows how genetic material is organized. Inside each cell, there's a thread-like structure called chromatin. When we zoom in on it, we can see chromosomes. At the center of these chromosomes is the DNA double helix. It looks like a twisted ladder. DNA is coiled up to fit inside these chromosomes and carries our genetic information. Each strand of DNA has many genes. Genes are small sections that give instructions to make proteins. These proteins do most of the important jobs in our body. Now that we understand what nucleotides are, let's take a closer look at their structure. We'll go through it step by step and see how these basic units connect and build up to form the full structure of DNA. First of all, we'll start by looking at the nitrogenous base. This base is a key part of the nucleotide's core structure. It connects directly to a sugar molecule known as a pentose sugar. So, the basic core of a nucleotide includes this nitrogenous base and the sugar. Now, there are 4 types of nitrogenous bases that we commonly see in DNA: adenine, thymine, cytosine, and guanine. Each of these bases plays a very important role in the genetic code, and that's because they pair up in a very specific way, as adenine always pairs with thymine, and guanine always pairs with cytosine. This specific pairing helps DNA carry and pass on genetic information accurately. And talking about how a nucleotide is formed. A nucleotide is completed when a phosphate group attaches to the pentose sugar. This forms what we call a phosphoribose structure. Finally, the phosphate group bonds with the sugar through what's known as a phosphodiester bond. This bond is essential because it links one nucleotide to the next, forming a long chain. And it is this chain that creates the backbone of the DNA molecule. So in summary, a nucleotide is made of 3 main parts: a nitrogenous base, a sugar, and a phosphate group, each playing a critical role in building and stabilizing the DNA structure. Now, let's take a closer look at the DNA double helix structure formed. As we've already seen, DNA is made up of two long chains of nucleotides. These two strands twist around each other, forming what we call a double helix. You can imagine it like a twisted ladder. The sides of this ladder are made up of the sugar-phosphate backbone. This backbone is formed when the sugar of one nucleotide connects to the phosphate group of the next, and this pattern continues all the way down the strand. It provides strength and structure to the DNA molecule and runs along both sides of the double helix, holding everything together. Now, if we look inside this twisted ladder, we see the rungs. These are made up of the nitrogenous bases from each strand. The bases from one strand pair with the bases on the opposite strand in a very specific way. As we already discussed, adenine pairs with thymine, and guanine pairs with cytosine. These base pairs are held together by hydrogen bonds. Hydrogen bonds are not too weak and not too strong. They are just right, they hold the two strands together, but they also allow the strands to separate easily when needed. This is very important during processes like DNA replication, where the strands need to unzip to make copies of the DNA. So overall, this beautiful double helix shape of DNA is not just for appearance. It makes DNA strong, flexible, and functional. It helps protect the genetic code, allows for easy copying during cell division, and makes sure the instructions for life are stored safely inside every cell. Alright, so let's do a quick recap to sum everything up. A nucleotide is considered a biological molecule because it's a naturally occurring chemical compound. It is found in all living organisms, and it's essential for life. It helps carry genetic information and takes part in many important cell processes. Now that we understand this, let's go back to how DNA is built, starting from its smallest parts. If we break down DNA, here's what we find. First, we see the DNA has a double helix shape. It looks like a twisted ladder. That's the full structure we call DNA. If we untwist this double helix, we get two long strands, and these strands are made of repeating units. These units are called nucleotides. Now, each nucleotide is made of three parts: a phosphate group, a sugar, known as pentose sugar, and a nitrogenous base. If we zoom in further, we see that the nitrogenous bases on opposite strands always pair up in a specific way. Adenine pairs with thymine, using 2 hydrogen bonds, and guanine pairs with cytosine, using 3 hydrogen bonds. These base pairs make the rungs of the DNA ladder, while the sugar and phosphate form the sides of the ladder. Together, this forms what we call the sugar-phosphate backbone. And that's what makes the nucleotide such an important biological molecule, because it's the basic building block of DNA. It plays a central role in storing and passing on the instructions for life.